right, everybody. Good morning. I hope you had a great weekend. It's 9:04, so we're going to need to get started early. So open up a session in catalytic learning. The question is, what is the number one cause of death?
The leading cause of death in the U.S. every year is heart failure. Each year, about 600,000 people die. This is about the population of Portland, Oregon. Heart failure occurs when your heart becomes starved for energy, or ATP. It cannot pump enough blood and oxygen to meet the demands of the body. The primary issues in heart failure occur in the left ventricle, which is the chamber of the heart that pumps blood all around the body, and therefore bears the heaviest load of the chambers in the heart. It was once thought that heart failure resulted from the reduced function of the electron transport chain in mitochondria within heart tissue. The electron transport chain is just one step within the entire process of cellular respiration, which produces energy for the cell. It begins with glycolysis, where a series of reactions break a glucose molecule down into two molecules of pyruvate. The pyruvate molecules then get turned into acetyl-CoA, from where two carbons from the acetyl-CoA are added to the citric acid cycle. The citric acid cycle is a cycle of reactions that produce significant amounts of NADH and FADH2. These molecules act as electron donors to the electron transport chain, which uses these electrons to pump protons across a membrane. This resultant proton gradient is what is then used to create ATP via ATP synthase. However, a new study presented in the Journal of Molecular and Cellular Cardiology claims differently, though. These scientists claim that the previous studies, which used analogs for left ventricular tissue, such as animal hearts or human heart tissue from other areas of the heart, were flawed for not using left ventricular tissue. They claim that the mitochondria in the left ventricle fully maintain their functionality during heart failure. The tissue came from patients who are already undergoing various medical procedures where part of the left ventricular tissue was being removed or undergoing heart replacement surgery. To test their functionality, the mitochondria are isolated using a centrifuge and were suspended in a buffer solution containing one of four substrates: glutamate malate, pyruvate malate, palmitoyl carnitine malate, and succinate, all of which are involved in oxidative phosphorylation. The respiration of the mitochondria was then measured through their consumption of oxygen at four states: the mitochondria alone, the mitochondria with the substrate, the mitochondria, substrate, and ADP, and mitochondria, substrate, and FCCP, which acts to limit respiration. By this point, most of the ADP had been converted into ATP. The third state is like flooring the gas pedal on your car and trying to find the top speed. A high state 3 rate indicates the mitochondria are functioning at high capacity, and a low state 4 rate indicates that the mitochondria are functioning with efficiency within the electron transport chain. Their study found that not only did the mitochondria from patients with heart failure maintain their functionality when isolated, but that their respiratory control ratios, RCR, which is the ratio of respiration at state 3 to state 4, were significantly higher for all substrates other than succinate than non-heart failure mitochondria. The respiratory control ratio is used for comparison because a high RCR means high state 3 and low state 4 respiration rates, which means the mitochondria are operating very well. The respiratory function was also significantly greater in the mitochondria taken from obese patients than non-obese patients, and it is possible that the mitochondria within the obese patients adapted to the environment where there was a higher amount of lipids present. The scientists believe that this increase in RCR is due to reduced state 4 rates. The scientists also took RNA from the tissue samples and transcribed them into cDNA to determine the expression of genes responsible for metabolism. They found that the expression of genes that regulate fatty acid, glucose, and pyruvate metabolism were downregulated in patients with heart failure. So, these pathways would not produce as much of the necessary substrates to metabolize them. The only gene they analyzed that was not downregulated was the gene for fatty acid uptake. While this does not prove anything, it suggests that the lack of energy in heart tissue during heart failure is not due to the reduced functioning of the electron transport chain, as previously thought, but a reduction in the production of the metabolic pathway. So, the study showed that mitochondria are not irreparably damaged in heart failure, but that something else is the cause for a lack of energy in the heart. The scientists say that future studies should focus on whether the starvation is caused by factors that limit the delivery of substrate to the mitochondria, a lack of mitochondria, or another factor limiting mitochondria function within the heart. So in conclusion, wait, did you really sleep through that amazing lesson? I've got to leave. I've got an appointment with a unicorn and a leprechaun after this. You guys are the worst.